My name is Maritza Sanchez, and I have no conflicts of interest to disclose at this time. What is the human small bowel, and why is it important? The human small bowel is a hollow tubular structure with an average adult length of 22 feet, making it the longest part of the gastrointestinal tract. The small bowel is critical because it's the primary site of nutrient absorption and maintains a tightly controlled environment. It was previously believed to be a low biomass and low complexity. It is related to many processes such as food digestion, absorption, and control of metabolic hormones, the immune system, and other very important functions not seen in the large bowel. When the balance is disrupted, it can lead to conditions like small intestinal bacterial overgrowth, otherwise known as SIBO, resulting in malabsorption and other gastrointestinal symptoms. Small intestinal bacterial overgrowth was believed to be the excessive growth of overall bacteria in the small intestine that led to disruption in digestion, nutrient absorption, and gastrointestinal symptoms. SIBO was historically thought to be the excessive growth of bacteria moving from the colon to the small intestine. SIBO is now recognized as a complex small bowel microbiome disorder, not just bacterial migration from the colon. Through our research, we have made many advances in understanding SIBO. We have improved breast testing culture. We progressed to 16S sequencing, as well as quantitative 16S sequencing and shotgun sequencing. There are two main ways of diagnostic testing for SIBO. The indirect method, being breath testing. The other, being invasive, is direct testing, where subjects undergo an upper endoscopy, or EGD, where small intestinal aspirates are collected and cultured on MacConkey agar. The North American consensus for diagnosing SIBO are bacterial cultures that are greater than or equal to 10 to the 3 colony-forming units per mil. MacConkey agar is a highly selective medium designed to isolate members of the Proteobacteria phylum, which are mostly gram-negative, fast-growing bacilli, and strong sugar fermenters. In the human body, we don't have an anaerobic system. It's actually a mixed environment. That distinction is exactly why MacConkey agar is used in diagnosing SIBO. This taxonomic profile demonstrates what we will see in a normal subject with less than 10 to the 3 CFU per mil. As you can see, it exhibits a relatively diverse microbial community, as opposed to what we see in SIBO subjects with greater than or equal to 10 to the 3 CFU per mil, demonstrating a marked dominance of Proteobacteria, particularly Endobacteriaceae, indicating overgrowth of specific gram-negative organisms. Sunburst taxonomic plots, built from the center being the domain, and build in layers of phylum, class, order, family, genus, and species. In history, this is the first shotgun sequencing of the human small intestine in SIBO. Shotgun sequencing captures all DNA within a sample, allowing comprehensive characterization of the microbiome. Our findings show that SIBO is predominantly composed of 18% of Klebsiella pneumoniae, 28% of Escherichia coli, and together making a total of 46% of the whole duodenal microbiome. Metagenomic sequencing has helped us identify the key players in SIBO, but interestingly, we haven't fully characterized these organisms when grown on MacConkey agar. Therefore, we aim to comprehensively characterize the species, strains, and genomic features of bacteria isolated from MacConkey agar cultures of duodenal aspirates in SIBO using whole genomic studies. For our methodology, samples were aspirated using a sterile double lumen catheter. Duodenal samples were collected and transported to the lab and cultured on MacConkey agar. Those meeting the SIBO threshold were swabbed with a DNase-RNase-free swab. Swabs were frozen and loaded, DNA was extracted and loaded on the NovaSeq. Microbial identification was performed using a Kraken 2 with a high confidence score of 0.9. Downstream analysis included genome assembly, alignment, and strain level characterization using an average nucleotide identity, otherwise known as ANI, and an alignment percentage, AP. Finally, antibiotic-resistant genes were assessed using a shortbread IAR database. Here we show the subject demographics for those with growth on MacConkey agar. Importantly, all subjects demonstrated bacterial growth, meaning the From our cohort, we saw that Klebsiella species and E. coli are the most prevalent species in SIBO. Bacterial colonies from 7 MacConkey plates were sequenced, yielding an average of 3 billion reads per plate. Consistently with prior reports, Klebsiella and E. coli were the most prevalent species from 6 out of the 7 plates. 6 of the 7 samples had either Klebsiella, E. coli. Here we wanted to see how similar E. coli genomes from SIBO are compared to a well-characterized reference strain, E. coli K12. On the left, we see whole genomic alignments between SIBO isolates, MacConkey 6, and MacConkey 7 and E. coli K12. Each color block represents a conservative genomic region, and connecting lines show how those regions align across genomes. What we observe is that these genomes are largely conserved, indicating a strong similarity to K12. To quantify this, we use an average nucleotide identity, or ANI. As shown here, ANI values that are around 98 to 99%, confirming that these SIBO isolates are highly similar at sequence level. However, when we look at alignable percentage, we see lower values around 76 to 80%, which tells us that not all genomic regions are shared. So while the shared DNA is highly similar, there are portions of the genome that are either missing, rearranged, or uniquely acquired. And when we further characterize these genomes, we see that E. coli strains carry genomic traits associated with pathogenic strains. Together, this suggests that although SIBO-associated E. coli are highly similar to commensal strains like K12, they harbor additional genetic modifications that may have functional or clinical relevance, though their exact impact remains unknown. For Klebsiella pneumoniae, we see a very different pattern across independent SIBO cultures. Klebsiella genomes are highly conserved. The genomic alignment shows strong similarity between isolates, and this is further supported by a high ANI value as shown below. This suggests that unlike E. coli, Klebsiella and SIBO may represent a more uniform population of closely related strains. Overall, this points towards a consistent genomic signature for Klebsiella across all patients. After identifying Klebsiella pneumoniae as a dominant organism in SIBO, we next asked whether these strains carry antibiotic-resistant features. We see the distribution of antibiotic-related virulence genes across our samples. Notably, these genes are consistently present across multiple isolates. When we look more closely, we identify resistant mechanisms spanning multiple antibiotic classes, including beta-lactams, cephalosporins, and fluoroquinolones. Importantly, we also detected mutations in OMPK37, a porin involved in antibiotic permeability. Alterations in this gene are known to reduce susceptibility to carbapenems. Together, these findings suggest that Klebsiella pneumoniae in SIBO is not only present, but may carry clinically relevant resistant traits. We next performed a similar analysis for E. coli to evaluate antibiotic-resistant genes profiles. As shown here, E. coli isolates also carry a wide range of antibiotic-related virulence genes across multiple classes. In one of the E. coli plates, we had all the expected resistant gene profiles to antibiotics. In the first plate, we didn't find any further resistance. However, on our second plate, we did find one mutation that's related to ciprofloxacin resistance. Some of the limitations included having a small sample size. However, deep sequencing was performed on dozens of colonies. Second, swabs were collected from the entire plate rather than from individual colonies. Lastly, we did not study SIBO-negative subjects yet. In conclusion, there are 4 key takeaways from our study. First, Klebsiella species and Escherichia coli are the most prevalent microbes in subjects with greater than or 10 to 3 CFU per mil on MacConkey Agar. Second, E. coli genomes are highly similar to K12, but with additional genetic modifications. In contrast, Klebsiella pneumoniae showed moderate genomic variation. And more importantly, across both organisms, antimicrobial resistance were commonly observed. Going forward, we plan to focus on the following areas by identifying the key E. coli and Klebsiella strains involved in SIBO, we can now begin to investigate their functional potential, such as mucosa adherence, metabolic activity, and hydrogen sulfide production. Importantly, understanding these strain level differences may open the door to a more targeted therapeutic approach. Thank you so much for your time.